All right, good morning or good afternoon or good evening to you all, wherever you are in the world today. I want to thank you for joining us for this presentation today. Uh, we're going to be presenting to you EndoPrep from Predictive Oncology sample treatment with PYROSTAR ES-F plate. This is a collaboration between my company, Fujifilm Wako Chemicals, and Predictive Oncology. And I want to welcome as well Chrysanthi and David. We'll be introducing ourselves in a moment. But we want to show you how Fujifilm and Predictive Oncology can aid in the testing of your drug products and safety efforts. I'm going to give just a brief introduction to myself to start off. My name is Timothy Francis. I'm the Senior Technical Specialist of the LAL division here at Fujifilm Wako Chemicals USA. I provide much of the training, instruction, technical management, as well as performing various studies for products that we would like to collaborate with as well. I come into this role with my undergrad degree in microbiology, as well as the curriculum and instruction in science master's degree. I'm going to pass it on now to Chrysanthi Weaver, and she'll present for us. She'll introduce herself, and then David will provide his introduction and then introduction to this overall study.
Thank you, Timothy. Hi, everyone. My name is Chrysanthi Weaver. I am a Director of Research and Development at the Biologics Division of Predictive Oncology. At Predictive, I manage the protein formulation development projects, R&D, as well as the endotoxin related products. My background is I have a bachelor and PhD in chemical engineering. So now I'm gonna let Dr. David Johnson to introduce himself.
I'm David Johnson, Director of Engineering for Biologics at Predictive Oncology. I have over 10 years experience supporting preclinical drug development and a PhD in biomedical engineering. And I'm going to go ahead and start our outline. So first of all, what we've done here is independently evaluated the use of EndoPrep sample treatment with PYROSTAR ES-F plate to detect endotoxin in the presence of 4 off-the-shelf biologics. And we've developed an example protocol to aid the use of products together. And here's a brief outline. For the introduction, we're going to be talking about endotoxin testing and the purpose of each product. With materials and methods, we'll detail sample preparation, testing, and result interpretation. And then we will present the results with 4 biologics and provide some conclusions and recommendations. So the goal here is the accurate detection of this molecule, lipopolysaccharide or endotoxin. It's a ubiquitous and potent pyrogen, and because it elicits a strong immune response, it is regulated by pharmaceutical quality control systems such as U.S. Pharmacopoeia and the AAMI. Accurate detection of this molecule is a challenge. It's found in the membrane of gram-negative bacteria, and proteins commonly interfere with testing. So we have our 2 products, PYROSTAR ES-F plate, which is an FDA-approved endotoxin detection kit, and EndoPrep, which is designed to reduce protein interference. It is a pre-sample treatment for the test. And just to reiterate how difficult it is to detect endotoxin, this is the list of common interferences and mitigations provided in USP 1085. And you'll see where the 2 products come in. The PYROSTAR is designed to prevent glucan interference, while the EndoPrep has LAL compatible protease to prevent protein interference. And the protocol that we've developed also includes dilution steps and back calculation of the product endotoxin levels. So you get additional mitigations in that process. So let's see. Now I'm going to turn it over to Timothy to go over the bacterial endotoxin test and LAL protein cascade.
Yeah, thank you so much, David. So the bacterial endotoxin test originally was developed as an alternative to pyrogen testing. Pyrogen testing, of course, is an in vivo test in which the pharmaceuticals are injected into living animals to directly measure a pyrogenic response. The bacterial endotoxin that David was just discussing, of course, is the most potent pyrogen and the most resilient pyrogen. So really it is the molecule that is detected with pyrogen testing. Of course, there are tests that require full pyrogen testing, the non-endotoxin pyrogens as well, but in most cases, it's endotoxin that are in those pharmaceuticals that cannot be necessarily guaranteed to be mitigated through manufacturing processes, and so they need to be tested for. So that's the overview, a brief overview of the history of the bacterial endotoxin test. Here in the United States, the USP sets the limits and the guidance. That's Chapter 85 for pharmaceuticals. They also have Chapter 161, but in reality, it's the AAMI that provides limits and guidance for medical devices. And then the USP provides monographs that will provide specific endotoxin limits for pharmaceuticals, as well as the AAMI different limits for medical devices. The reagent currently that is used for bacterial endotoxin testing is Limulus amoebocyte lysate. Of course, we are moving on to recombinant methods as well, but they're all based on the same principle, the same endotoxin cascade as you're seeing here on the right of your screen. Basically, what naturally occurs in the hemolymph of Limulus polyphemus is 3 proteins that are a part of a cascade that ultimately lead to the formation of coagulin from coagulagen. So endotoxin specifically is binding to that factor C. That factor C activates and cleaves factor B, which in turn cleaves proclotting enzyme to our clotting enzyme, which will, in the hemolymph itself, create the coagulation by coagulin. There are also reagents that have the chromogenic substrate that is added to create a higher response. But it's this basic cascade pathway that is present in all forms of LAL or synthetically derived reagents. There's also factor G. David mentioned the interference that can be caused in the bacterial endotoxin test from glucans. Our PYROSTAR reagent comes with large amounts of curdlan added to the reagent, which actually binds to factor G and keeps it from activating in the presence of glucans in the molecules, thereby making the reagent endotoxin specific. However, it's this cascade that gives such high sensitivities to endotoxin that is particularly vulnerable to protein interference, because those proteins can come in and either inhibit, prevent part of this reaction from occurring, or enhance, cleave some of those proteins and provide reaction. So proteins, in a sense, provide the most troublesome portion of interference with the bacterial endotoxin test. Of course, there are mitigating factors such as dilution and heat treatment that David mentioned, but there are some cases where the interference persists. So it's the EndoPrep product specifically that we want to use with our reagent to provide that even more mitigation of protein interference. We're going to move on to the next slide now. And that's the brief introduction to the overview, kind of the science of the the product. Now we're going to look at the materials and methods of the study that we performed. So I guess it was about a year ago now that we initiated this study, and we brought the products into both of our labs, and we performed the study in duplicate one, at least at duplicate sites. The product that we used was PYROSTAR ES-F plate. We specifically use this product because among our reagents, this is the reagent that has the, in a sense, the highest, so the least sensitive of lambdas, that lowest amount of endotoxin detection. So in a case, this is the reagent that would be most prone to protein interference, and then of course our other reagents would work even better with the kits that were used. So we use PYROSTAR ES-F plate, in addition to the fact that microplate readers are nearly ubiquitous in the lab. So nearly any microplate reader, as long as it's incubating, it can measure absorbance at 405 nanometers, and is able to perform kinetic measurements, will comply with our package inserts and be able to run a fully compliant FDA licensed assay with that. Our kits themselves come with control standard endotoxin. This control standard endotoxin is reconstituted according to reference potency found during LAL manufacture. So when we manufacture the reagent on site, we use reference standard endotoxin from the USP. And this reference standard endotoxin is used to create the validation curves. What then is used is our control standard endotoxin is calibrated based on the performance of that. So that's why it's lot to lot specific, but this provides a calibrated standard. When reconstituted according to the package insert, the vial is 1,000 EU per milliliters. Of course, with endotoxin sensitivities in 1, 0.5, 0.25, 0.015 endotoxin units per milliliter, this needs to be diluted down. So on the right of the slide, I show the serial dilutions that we use to bring this down. And in this case, we use a standard curve of 10-fold dilutions. Of course, other dilutions are perfectly acceptable and can be used. But in this case, this generally illustrates the standard curves that were used from the 1,000 to 100, 10, 1, 0.1, and then finally 0.01 EU per ml concentrations, using serial dilutions of 0.2 milliliters of whatever standard endotoxin concentration was used before, diluted in 1.8 milliliters of lysate reagent water. The LAL itself is reconstituted according to the label on the vials. In this case, that's 2.0 milliliters. And then for the actual test itself, 1 part reagent is added to 1 part sample. So in this case, that's 50 microliters of reagent added to 50 microliters of sample. And that's all what is needed, as this is a kinetic technique. Once that's added for the entire assay, it's immediately placed in the reader, incubated at 37 degrees Celsius, and the readings immediately start. We're going to go to the next slide now. What directly we are measuring as a metric of interference is recovery rate, in terms of measured value divided by reference value or actual value. Our criteria for acceptance is the pharmacopeially required 50 to 200% recovery. And I have this illustration here just to bring together the whole picture of what is taking place. So you can see on this graph, I have my standard curve. You can see on the x-axis, I have log endotoxin concentration. Basically, these 3 points that you're seeing here that are at -2, -1, and 0, that's the log. That's the exponent of the base 10. So really, this is from left to right. The first point is at a 0.01. The midpoint is at 0.1, and then the point on the right is at 1 EU per ml concentrations. And then the y-axis shows for us the log of activation time in minutes. So you'll notice I have off of the y-axis referenced around 1.2 and around 1.4. Once again, that's the logarithm. That really corresponds to around 12 minutes and around 16 minutes. So if between 12 minutes, 16-minute variation in activation time corresponds then, as we bring that across to the standard curve, to a 50 to 200% recovery. So in this case, the 50% recovery would be around a 0.05 EU per ml, and then 200% is 0.2 EU per ml. So that just shows the variation in activation time leading to variations in the standard curve concentrations that are allowed. So that protein interference that I was mentioning will cause variations in activation time one way or another. And based on the standard curve recovery, that recovery value will be compared to the actual value to give a percent recovery. And on our recovery slide, we'll have that equation for you to reference. So that gives the idea. We are creating, basically, we're spiking our samples to the midpoint of the standard curve. So the final positive product control, in a sense, has endotoxin added at the midpoint of the standard curve. And then by measuring changes in activation time, gives us an idea of that recovery. And as long as we're within a 50 to 200% recovery, we have, by USP 85, passed the test for interference. However, proteins will create inhibition, in which case our recoveries are shifted over to the left of the curve, or enhancement, where our recoveries are shifted to the right in terms of endotoxin concentration versus actual values. So I'm going to pass it now on to Chrysanthi to describe the preparation of the EndoPrep kits.
Thank you, Timothy. So on this slide, we show you a picture of our kit, an EndoPrep kit. This kit contains a digestion buffer vial and a lyophilized protease vial. So typically, prior to use of the kit, the protease vial has to be, the protease has to be reconstituted using a meal of the digestion buffer to form the protease solutions. In the form of solution, the protease solution is typically stable to be stored at 4C for a period of 1 month. The shelf life of the brand new kit and brand new unused kit of the EndoPrep kit is 1 year at 4C. So for the next couple of slides, I will describe a little bit more details of the materials and methods that we use in our collaborative work of digestion experiments using EndoPrep kit prior detection with the PYROSTAR LAL assay. Next. So aside from the both kits, the PYROSTAR and the EndoPrep kits that we use, all the experiments that we perform in this work, they are all prepared and performed in pyrogen-free dilution tubes. We use LAL reagent water to perform any sample dilutions. And as Timothy mentioned, that the LAL experiments, they were performed in 96 well microplate. In this case, we used the BioClean plate from Fujifilm. As for the detection, we can use any brand of plate reader that is capable of performing kinetic turbidimetric assay at 405 nanometer. For this work, we Tested several proteins and peptides. We obtained commercially available bovine serum albumin or BSA, bovine IgG, and human hemoglobin. For the sample of peptide, we tested a Sushi-3 peptide. Sushi-3 peptide is a short peptide around 3,000 Dalton molecular weight that binds strongly to endotoxins such that it inhibits endotoxin detection in LL assay or in any chromogenic assay. So next slide. So now in this slide, we'll go over a little bit more of the experimental setup on how to we set up the samples as well as the neutralization method using the EndoPrep prior LAL experiment. So for each of the protein or peptide that we tested, we prepare 4 set of samples in pyrogen-free tubes. So 2 of the tubes are labeled PPC or positive product control. PPC tubes contain protein solution that are spiked with a known concentration of endotoxin. One of the PPC tube is treated with EndoPrep, it's hence labeled treated PPC tube, and the other tube is untreated. That means we did not add any protease solution. Instead, we add the same volume of a digestion buffer. Likewise, for the other 2 tubes, we label them NPC or negative product control. NPC tubes only contain a protein solution without endotoxin spike. And similar to the PPC tubes, one of the tube is treated with EndoPrep while the other tube is not. So basically, with these 4 tubes, we're basically preparing an experiment that we can compare the endotoxin recovery in the presence of protein and in the presence of EndoPrep versus without EndoPrep treatment. And once we set up the samples, then I'll go over the neutralization procedure with EndoPrep. So this method is described in our the user manual that comes with the EndoPrep, as well as protocol template that comes with our white paper that we're presenting in this webinar. So basically, we mix about 30 microliter of the protease solution with 270 microliter of protein sample. Once we set all these tubes, we seal these tubes with parafilm and then incubate the tubes in water bath at 37 degrees C for about an hour. So after incubation, we took these tubes out of the water bath and then we dilute the samples 1 to 100 with LAL reagent water or endotoxin-free water. So then we add about 50 microliter of each sample into microwave plate plus 50 microliter of the LAL reagent to start the turbidimetric assay in plate reader. So now I'm going to pass the presentation back to Timothy to explain some of the interesting results that we obtained from our study.
Yeah, thank you so much, Krisanthi. So, of course, we did this at both of our sites. Each of these different proteins was tested. As Krisanthi mentioned, we tested the sample in PPC. In each case, the only difference was we would test the sample in PPC, then in comparison with an untreated sample in PPC and then treated sample in PPC. So the untreated sample and positive product control would go through the exact same dilution and as well as the heating that we underwent with the treated sample. The only difference is that EndoPrep digestive enzyme was not added. And these are the results that we obtained from this. So here we have, and actually the colors I believe have not translated through, but on the left side of each portion we have the without EndoPrep portion colors for that, the recovery rate. You can see on the y-axis 50 to 200% for there, and then each bar on the right shows the results after the EndoPrep treatment. So first of all, hemoglobin, notable recovery improvement was measured. You can see there we had recovery, very low recovery for the hemoglobin, and then after treatment, basically the inhibition was removed. We did get levels of enhancement, as you can see, and the replicates that we were getting, basically some of them were falling below the 200% recovery, some of them above. That was caused not by any protein, I believe, as a part of it, but just hemoglobin. Of course, it has that chromophore. It's very highly colored, and that was interfering with the absorbance portion of the assay. Here we go. Now we got the colors up for that, as you can see the distinction between those. So the I believe in this case the EndoPrep treatment did fantastic in removing that inhibition, and just further dilution or a chromogenic assay may be needed to remove those technical interference issues caused by the color. So hemoglobin, can't necessarily specifically recommend the procedure that we used, but we saw marked recovery with that. For the BSA, the bovine serum albumin, this sample in and of itself at that dilution, at that 100-fold dilution, already has mitigated the interference caused by the proteins in those samples. So in this case, the treatment, I believe, just in a sense added extra noise to the assay, as you can see there, and you can see the recovery rates. Of course, this is a it's a highly it's serum. It's highly viscous. There are probably other technical issues with that sample that could be overcome by further dilution. But you can see as well there is marked recovery increase in the sense of this treatment. However, this treatment wouldn't necessarily be suitable, as the recovery is already at the 100% range. So these are the two samples that we tested that we saw the effects of the treatment, but didn't necessarily with the procedures we used provide results that fully met the pharmacopoeial requirements. I'm going to pass it on now to Chrysanthi to show the proteins that we did have very good results in.
Thank you, Timothy. So like he mentioned, now we look at the sushi-3 peptide and the IgG protein. So if you can see on the plot that in the presence of the sushi-3 peptide or IgG, the endotoxin treatment is the recoveries of the endotoxin from the LAL detection is very significantly inhibited, as shown by the percent recoveries that is way below 50%. However, after EndoPrep treatment, the percent recovery range increases to close to 100%, suggesting that the interfering effect by the sushi-3 peptide or the IgG in the solution is greatly reduced by the protease in the EndoPrep kit. Suggests likely the effect is due to the freeing effect of the protease in the solution, such that the endotoxin is now freely soluble in the solution to be properly detected by the LAL assay. So the variety of results that we see here suggested that EndoPrep kit can definitely be used to reduce interference on a variety of protein and peptides. And the different effects and percent recovery that we see here suggest that the use of EndoPrep kit along with the LAL assay has to be validated for each of the protein of your interest in your company. So now I'm gonna pass the presentation to Dr. David Johnson to provide and offer a conclusion of our study, as well as a recommendation moving forward.
Thank you, Santi. So I'm just going to go over some conclusions and recommendations. First of all, the PYROSTAR ES-F plate is FDA-approved LAL assay designed to overcome glucan interference, and the EndoPrep is designed to help overcome protein interference and endotoxin detection tests. Now, the USP provides endotoxin limits and guidance for pharmaceuticals, where this test is applicable, and your particular samples must be validated. Your particular products must be individually validated according to this USP guidance, or in the case across the world, your regional quality control standards. So we provided this example, this study, as an example of how to use the products together, and we've also provided an example protocol separately to guide development of this validation process according to USP guidance, which, due to harmonization across multiple quality control systems, should be very applicable to your region. And that's it. Here are our emails, and we have the white paper and example protocol online. We will be providing the slide deck as PDF for you to review, and as we'll also be providing the white paper and example protocol on the webinar site that you went to. And with that, we'll open it up to questions. If anybody has questions from the chat.
Yeah. Yeah, and if you have questions now, please leave those in the chat. There's also the Q&A portion, I believe, as well. You can leave them on either, but I'm looking at the chat right now. I see one question, and David and Chrysanthi, if okay with you, I'll go ahead and take this question since it refers directly to the LAL reagent. The question is there any chance of stopping gel clot, turbidimetric, and chromogenic by recombinant in the near future? So this question refers to the recombinant reagents, the synthetic alternatives that are coming out. Of course, they're in the process of validation by the various pharmacopoeias. We don't have harmonization yet, so the guidance in Europe is not necessarily the same as the guidance in the U.S. versus guidance in Japan. But what is the case, however, is that this protein treatment, the EndoPrep treatment, regardless of endotoxin test, will apply for the sample, because this is not it's not a buffer that's being added to the reagent. It's not a something added to the reagent itself. It's a sample treatment that is used. So the sample can be treated, and what it's literally doing is it's digesting, it's lysing those proteins and removing the interference of that. So if there is protein interference in the sample, this pre-treatment will remove that interference, keep the endotoxin in the sample so that way it can be treated after. All right, and then second question here is, hi, wouldn't one expect lower spike recovery with sushi peptides as they are known to bind LPS? How does your reagent act upon the endotoxin? So yeah, and actually, David, if you go back to that slide showing the sushi recovery, of course, the sushi regions of factor C specifically are those portions that naturally occur that from the the proteins. So here, predictive oncology, they use, I believe, correct me if I'm wrong in any statement I made, but you've manufactured basically this protein to bind specifically to that endotoxin based off of those regions. So you can see untreated without EndoPrep, we saw great amounts of inhibition. You can see the sample there without EndoPrep, the recovery rates. And then with the EndoPrep treatment, that digestion brought the recovery within the allowable range. So of course this includes spikes. The LPS was added and then treated. So we're able to measure that with that treatment. After that treatment has taken place, we are recovering the amount of endotoxin that we added as a part of the sample spike. Any portion you would like to add to that question, Chrysanthi or David?
I think that was a good summary.
Yeah. That's good.
All right, and then next question coming here. Why does CSE originate from E. coli? I'll take this question again, as once again it refers to our CSE product. So the really it's the LPS is really what we're looking for. A manufacturer could create CSE from pseudomonas. They could create CSE from any gram-negative molecule, and in fact, the original reference standard back in the 1980s was actually a pseudomonas LPS, not E. coli. Of course, the USP standardized E. coli as the 113 and I forget the exact serotype, but that's what we've been using since. We use E. coli for our LPS, for our CSE UKT. B is what we use, but there's no requirement that says the control standard endotoxin needs to come from E. coli, but the requirement is that it needs to be specifically calibrated to the reference standard endotoxin by the matching that takes place on site. And of course, the RSE is used for the standardization of all the tests throughout the world. And now we have, of course, the global harmonization between USP, EP, and JP produced endotoxin standards. All right. I see a couple other questions coming in here. I think I'll give this question either to Chrysanthi or David. Is your treatment solution yielding superior spike recovery compared to standard heating dilution protocols used to inactivate, inhibiting, or enhancing proteins in the sample?
Yeah, I'll take this. And the answer is yes. These samples were all treated with dilution and heat treatment in the negative, without inclusion of EndoPrep. So I'll go back to the results here. You see for example, the sushi 3 provided a significant inhibition of detection of the endotoxin that we spiked with, as Timothy reiterated, and this low recovery of endotoxin represents dilution and heating in similar method to the product that includes EndoPrep.
So let me add a little bit to that, the EndoPrep is very useful when the inhibition is caused by masking by the protein. In this case, probably the protein or peptide binds tightly or have a strong interaction with the endotoxin such that it, inhibits the detection method by the LL assay or chromatogenic assay.
And
So
to be clear,
sometimes when it binds tightly, then when you do a dilution, you might not see that effect.
yeah, exactly. And yeah, like Krisanti was saying, to be clear, you do not necessarily have to have EndoPrep if dilution and heating works. In the case of the Sushi 3 and this IgG, you do not get recovery in the 50 to 200% range with just dilution and heat treatment alone.
Yeah, it's if I can add, it's a tool in the toolbox. I mean, there's cases where there's a specific complex protein where you know, heat treatment, dilution down to the MVD aren't providing results, and you can see that with our recovery rates. Each of the proteins that were inhibited, we were already at 100-fold dilution, and you know, there's the option of, you know, using more expensive chromogenic reagent to try to increase the MVD, but sometimes that doesn't work. And really the options then, if as a last resort, are you have to move on to the MAT, or even employ the rabbit pyrogen test. So at that case scenario, this gives you an additional item in the toolbox. I think as well, not even when you're at that worst case scenario, but here it's allowing you to take the advantage of heat treatment and dilution and a digestion all at once to really combat some severe interference with the protein. So this is just one of those tools, hopefully a simple tool in that toolbox for overcoming protein interference in our samples that we're testing with the LAL assay, where you know, going to an alternative method really isn't desirable at that moment. This next question, is there any reference for CV percentage, and what is the limit? So CV, of course, percent coefficient of variation. There's no pharmacopely required limit for percent CV. Percent CV often is set when looking at reference standards. We like to have those, or control standard, the standard curve, we like to have those below 10%. Often with samples, the limit is set at 20 to 30%, but there's no specific requirement. Of course, percent CV is a measure of how consistent are your results. So higher percent CV values. Of course, if the one of those values is above a limit, then that becomes a limit. But really, that's an informational measure of how tightly, in a sense, your protocol is at providing consistent results.
And all of these depend very much on your specific drug product and the development of your own protocol for testing endotoxin according to standards.
All right. So we have one more question that came in. Hi, Wako lysate, once reconstituted with LRW, can be stored at 2 to 8 degrees Celsius for 8 hours, but other manufacturers claim for 24 hours at 2 to 8 Celsius. What is the reason behind it? Please clarify. Once again, this is a lysate reagent question. So the reason why our shelf life is set the way it is because we have a dual purpose reagent. The turbidimetric results allow for that. Of course, we provide vials that are have two milliliter quantities to, you know, hopefully bring you into the amount of sample that's required for that assay, as well as freezing can be used to save that for another date. So hopefully that, along with the cost advantages of our reagent, will mitigate the, you know, the difference in you get a whole day of use in 8 hours if you time that right, versus 24 hours for other samples. As other questions continue to come in, I'm going to ask David and Krisanti if I know we're going to send a email out to further to those that have registered, but what website, where should those that are watching go to if they want more information on your product specifically, but then also how your product works with ours?
I believe those links were provided in the preliminary notifications for the webinar. In 1 second, I will get them up quickly. Yeah, so there is a site for predictive oncology. It is predictive-oncology.com/Fujifilm, and that describes the collaboration and gives more information, the link to the webinar, where you'll be able to find the detailed documents.
Yeah, thank you. And thank you, Teresa, your colleague from Predictive Oncology. She just put that in the chat as well. So if any of you watching would like to see the further information, we'll send you the emails and follow up as well. But if you want to go to that directly, feel free to copy that, click that in the chat currently. I think, though, that we've answered the questions that have come through. If you have any further questions, David gave you the email, and if you could go back to that page, I'm currently seeing the conclusion and recommendations page. So you can email me directly. You can see timothy.francis@Fujifilm, or David or Krisanthi. You see their emails there. Feel free to email us directly with questions after this, and we'll be glad to answer any questions you have. And if you get the product, you can get the product from Predictive Oncology. Of course, you can get our product from us, as well as, and they can provide you the information on their distributors. But if you get the product, you get our reagent, please email us. We'll be glad to walk you through the protocol step by step. And we'd love to hear your results. If you have a troublesome sample, that this may be able to be of benefit for you in providing endotoxin recovery for that. So yeah, we'll get the other questions that come through by email. I'm not sure I 100% understand this question right here, so I'll follow you up by email with that one. And then, yeah, please email any other further questions that you have. But I want to say thank you to David, thank you to Krisanthi for presenting this with me. It's been a great time, actually. I've enjoyed working on this project with you over the past year and a half or so.
That's very enjoyable.
Likewise. Yep.
Mm-hmm. Thank you.
All right. Well, you all have a great day, and we'll be in touch if you have any questions. And thank you so much for joining.
Thank you.
Have a great day.